Hi guys, this is IGCSE paper 2 for 2020 May June. It's 0610, or actually 0610 slash 21. So let's have a look at the first one. So the diagram shows what happened in an experiment with plant seedlings. So which characteristic of living things made the seedlings grow towards the light? So this question, well, since it's number one, the topic is going to be the first topic, very first topic, called characteristics of the organisms. So when you think about this, there are plants, and they are making a respond towards the light. So the light is coming from the right side, and they are bending towards the light. This is actually this process or mechanism or phenomenon is called phototropism. So phototropism comes in topic 14. However, this question is rather simple. So what are these plants doing? It's basically it's sensing the light as stimulus and making a response. So the answer should be D, sensitivity, because sensitivity is the ability, or let's say it's a process of detecting stimulus and responding. So D is the answer. Of course, excretion is not the answer, because excretion means you are getting rid of the toxic materials. Nutrition is not the answer, because it's not. It doesn't actually say that it's photosynthesizing. Of course, with the presence of the light, plants will photosynthesize and make their own food. So things like glucose or glucose molecules and others. However, it's not showing that it's happening. So nutrition is not the answer, and respiration is not the answer as well, because it doesn't show, or it doesn't say anywhere that the plants are making energy, or it's making CO2 or water. So D is the only sensible answer. Number 2. A rat has a scientific name, or the scientific name, Rattus rattus. What do the two parts of this name refer to? So this is simple. Naming organisms always they are always based on this system called binomial system. So binomial system means bi means two, nomial means names. So you're giving two names to the organism, and the first one should be genus. Second one always should be species. So this question was quite easy one. So the answer should be a genus and species. Number 3. The diagram shows two guard cells. Which level shows the cell wall? So the guard cells are plant cells. And of course, the main difference between the plant cells and the animal cells are several things. So plant cells contain chloroplasts, whereas animal cells don't. They have cell walls. They also have large vacuoles. So let's have a look at this. So this is the nucleus, the biggest circular, or let's say circle-like structure. is always no nucleus. And these are chloroplasts. And C is a large vacuole, and B, okay, B looks like cell membrane. And D is, of course, the cell wall. The cell wall is rather thicker. It's actually much more thicker than the membrane. The membrane is very, very thin. It's not really stated in IGCSE. It's not actually learned. I mean, you guys never learn how thin the membranes are. You guys don't actually have to know how thin they are. So just know that these cell walls are quite thick. They're much thicker than these cell membranes, and cell membranes are always they are inside the structure, whole structure. The cell wall lies the furthest, so it's actually at the outermost part of the region of the cell, and the membrane is the next one. And then you get to see like a lot of cytoplasm here. So this is cytoplasm and other organelles inside the cytoplasm as well. So anyway, the bottom line is that the cell wall. Okay, and mind you, the cell walls are fully permeable. Whereas the cell membranes are partially permeable. Okay, awesome. Number 4. The diagrams show different levels of organization in a plant. So, which sequence shows the levels of organization in order from smallest to largest? So, this question is pretty simple if you can think about it. Number 4 is, of course, the smallest one because this is a root hair cell. And then, number 2, you can see that there are many cells gathered together. And that's going to be the next one, of course. And then they form what is called the roots. And of course, the roots are part of the tree. So the order should be 4, 2, 3, 1. 4, 2, 3, 1. So the answer should be A. Easy peasy. Number 5. Which diagram shows the appearance of a plant cell several minutes after it has been placed in a concentrated solution of sugar? So the question is asking what happens to the plant cell if it is placed in a solution of sugar. So when it says a high concentrated or concentrated solution of sugar, it means it has a lot of salt or sugar. Not salt, what am I saying? It's sugar. So it has a lot of sugar, which means it has relatively less water. So, relatively, the plant cell now has more water than the outside. So the water from inside the cell will go out of the cell by osmosis. You guys remember that the osmosis is movement of water molecules from high water potential to low water potential. That's osmosis through partially permeable membrane. So that is really, really important. So when we talk about osmosis, you have to say it's the movement of water molecules from high to low water potential through semi-permeable or partially permeable membrane. Okay, so if that happens, the plant cell will lose a lot of water, which means its membrane will become, let's say, flaccid or plasmolized. So when the plant cell loses a little bit of water, let's say lost a little bit of water, then it becomes flaccid. But when it loses a lot of water, then its membrane will become, let's say, shrunk. For plants, we actually call it plasmolized. So I would say A is plasmolized form of the plant cell. If it's flaccid, then I would say the plant cell will become like this. Maybe it has lost a little bit of water and the membrane got a little shrunk like this. So this is flaccid. But if many parts of the membrane become shrunk or it moves towards the center, then it's called plasmolized. So this is plasmolized. This happens when plant cells are placed in a concentrated solution of sugar or salt. Okay? Anyways, for this question, the answer should be A. Cool. Number 6. Plant cells are placed in a solution with a higher water potential than the plant cell contains. What will happen? So what will happen to this situation? Or in this situation, I mean. Okay, so plant cells are placed in a solution. Okay, so let's just draw it. So now plant cells are in a solution. And that solution has a higher water potential than the plant cell contents. So now there's more water outside the cell than the inside the cell. So as we already know, the osmosis happens when there's high water potential, or higher water potential, and there's also a lower water potential. So the water molecules will move from the higher water potential to lower water potential through a semi-permeable membrane. So since plant cells have the partially permeable membrane, or you can say semi-permeable membrane or semi-permeable membrane, you can say that the water will move from outside the cell to the inside the cell by osmosis, because it's going to move through the semi-permeable membrane. So the direction of the water movement is, of course, enters the cell, and the volume of vacuole. Now this is something that we have to talk about. So the plant cell has many organelles, just like any other cells. It does have nucleus, like DNAs, and raphiar, ribosomes, mitochondria, chloroplasts, and also it has a very large vacuole. So the function of the vacuole is to store water, and other waste products as well, like salts and stuff. So when the water goes inside the cell, it doesn't usually stay inside the cytoplasm. It goes inside the vacuole for the storage, or as a storage. So the volume of vacuole will of course increase because the water will go inside the vacuole, so the answer should be B. The direction of the water movement enters the cells, the volume of vacuole increases. Okay, so the answer is B. Number 7. The diagram shows a section of DNA with 4 bases identified on one strand. Which sequence of bases would be on the other strand, starting from the top? Okay, so there's a crab arrow. Maybe it's okay. Well, it doesn't actually make this good arrow like this. I'll just do it manually. Anyways, so we have to find the sequence of bases. For the DNA, there's a rule. The bases, certain bases will only pair. They only pair with a certain basis. So, for instance, A will always pair with T. C will always pair with G. So, and this is called complementary base pairing. Okay, so this is a rule, and that happens to conserve the gene sequences. We'll talk about that later, actually. I mean, you don't actually have to know about that in IGCSE. You guys should know about that maybe in AP, IB, A-level, and other boards, but not in IGCSE. So you can just memorize, let's say, this term called complementary base pairing. And the rule is that the A will always bind with or pair up with T, C with G. Okay, so G will pair up with C, A with T, G, A. So from top to the bottom, it's C, T, G, A. So the answer should be B. Number 8. Which food testing solution shows a positive result when it turns from blue to purple? So food testing. So there are several methods for food testing that you guys should know. And this one talks about the solution changing from blue, the color of the solution changing from blue to purple. So the solutions that are initially blue are Benedict's solution and Burette solution. And there's also another solution called DCPIP. This is the solution to test the presence of vitamin C. So it starts with blue color, and then if there's vitamin C present, then it's going to change its color to colorless, or CORLOR, less, whichever you prefer. So, in this case, it's saying that the color change happens from blue to purple, and that belongs to the Burette solution. So, the Burette solution tests the presence of protein. I'm not a big fan of term purple. I'd rather use the term violet, which is slightly different actually, but yeah. So the way that I remember this or memorize this is that it's got certain similar pronunciation. So Burette, violet. Okay, so Burette starts with B, so it's blue color, and then it changes into violet, which is similar to purple. So be red, violet. The Benedict solution changes from blue to other colors depending on how much of monosaccharides are there. So it tests the presence of monosaccharides or reducing sugars. They're the same things anyway. So, or you can call them simple sugars. So there are many terms to call the monosaccharides or simple sugars or reducing sugars. Anyways, so it changes the color from blue to brick red if there are many simple sugars, such as glucose molecules. If there are many glucose molecules, then the blue color will change to brick red. If there are less glucose molecules, then it's going to change into yellow, like orange. Actually, it's going to change into orange first. Not first, actually. If there are slightly less glucose molecules, then it's going to change this color into orange. And then if there's even more or even less glucose molecules, then it's going to change to yellow. If there's less than that, it's going to change to green. And then if there's if there's no glucose molecules, then it's going to remain as blue. So, yeah. So for this question, the answer should be B. All right. And as for the ethanol, it's alcohol. So the alcohol is used to check if there is any fat molecules. Iodine solution is used to check if there's any starch molecules. Okay. Okay, so the answer is B for this one. Thanks, guys. Number 9. What is the definition of an enzyme? So enzyme, this is very, let's say, very important concept. So the definition of the enzyme is very important to know. It's basically a protein. It's made of a protein. I always draw enzyme like this. And it binds with substrate, or the substrate binds to the enzyme's active site. This is called active site. And if the substrate fits into the active site, then the reaction will occur and it's going to produce products. So it's a protein first of all, and what it does is it increases the rate of reaction. So with that enzyme, the reaction will occur very, very slow. So without the enzyme, we can't actually live. So there are many enzymes within us that helps chemical reactions to occur very fast so we can function. And any substance that increases the rate of reaction is called catalyst. So for this question, the answer should be D. Enzyme is a protein that acts as a catalyst, which means it increases the rate of reaction. So it hastens the reaction, chemical reaction. Okay? So this definition is very, very important because when you encounter the questions regarding enzymes in long answer questions or short answer questions, like written questions, quite often you have to give the definition of the enzyme. So it actually becomes quite handy to get a few marks. Okay, so memorize it. It's very important. Oops. Not the rectangle, but the star. All right, thanks, guys. Number 10. An experiment is set up to investigate gas exchange in aquatic plants. The hydrogen carbonate indicator solution is orange at the start. Which color is it after 3 hours? So, hydrogen carbonate indicator is used in this question, in this experiment. And the aquatic plant is, of course, the plant that can live in the water or underwater, and it can photosynthesize in the water as well, which means it will use CO2. So it's gonna suck up this CO2 and use it for the photosynthesis. It produces oxygen in return and glucose molecules as well. So it says hydrogen carbonate indicator is orange at the start. So how is hydrogen carbonate indicator used? It's used to measure the presence of CO2, like how much CO2 is present. Is there many, like a lot of, not many, many molecules, or a lot of carbon dioxide molecules in the area or not? So it depends on the color change. The color change actually depends on how much CO2 is present. So if there's a lot of CO2, let's say a lot. There's like a few molecules, CO2 molecules. If there's less CO2, it's going to change the color into close to the purple. Okay? So that's like there's a nice, very nice picture or the diagram, which I can't really find at the moment, but I'll just probably just draw it for you. Okay, so if there's less CO2, the color will change into the purple, or closer to purple. So this is just like a rule. It just happens. If there's a lot of CO2 molecules, then the color will change into yellow. So this is something you guys should just memorize. So hydrogen carbonate indicator will change the color to yellow if there's a lot of CO2. It's going to change the color to purple if there's only few CO2 molecules. Okay? So this is just one of those things that in biology you just have to memorize without knowing why. There's of course reason for this, but you don't have to know why it's happening. At this stage, you guys are preparing for the exams and stuff, so just memorize this part, okay? Cool. Hey guys, it's number 11. The diagram shows how the rate of photosynthesis varies with light intensity. The four curves show different conditions of temperature and carbon dioxide concentration. What limits the rate of photosynthesis at point P? Here's the question here. What is wrong with the arrow? Okay, so it's actually talking about the limiting factor here. So you have to know the limiting factors of photosynthesis, and there are 3 limiting factors for the photosynthesis that you guys should know. First one is in light intensity, as written here. Second one is temperature. Third one is CO2 concentration. So in this graph, you can see that as the light intensity gets increased, the rate of photosynthesis also increases. But when you see the bottom one, the lowest line here, you can see at point P, the rate of photosynthesis is no longer increasing, although the light intensity is keep on increasing. Okay? And if the temperature gets increased, you can see the rate of photosynthesis increases a bit more than the bottom one. But however, in similar place, a similar level, the same level, actually not similar, but same, the rate of photosynthesis is no longer increasing as well. And if the concentration is increased from 0.1 to 0.2, you can see that the rate of photosynthesis is increasing. And if both temperature and the concentration gets increased, the rate of photosynthesis also increases in the same level. Okay? So, at P, light intensity is, or the increase or decrease in light intensity is doing nothing. So, the light intensity is not the limiting factor at P, because the limiting factor is basically, you guys actually have to know the definition of the limiting factor. So it's a factor that limits the reaction rate in any life process. Okay? So it's the factor that limits the reaction rate. So this definition is, in my opinion, is quite ambiguous and quite confusing. So I actually like to explain in different terms. So limiting factors are something, or they're factors that does have an effect on the life process. So at P, you can see that the light intensity will increase or decrease. The light intensity is doing nothing on the rate of photosynthesis. It's not affecting much. It's actually not affecting at all. But the others, like the temperature and the concentration of the CO2, does have an effect. So if you change the temperature to 30 degrees Celsius, you can see the rate of photosynthesis gets increased. You can also see that by increasing the concentration of carbon dioxide, the rate of photosynthesis gets increased. If you change both temperature and the concentration, like increasing them, you can see that the rate of photosynthesis also increases again. So the limiting factor at P is not the light intensity, but both carbon dioxide concentration and the temperature, because they both affect the rate of photosynthesis by increasing or maybe even decreasing as well. Okay? So the answer for this one is C. For this video, I actually spent quite a lot on explaining the limiting factor, because it has always been quite ambiguous and quite confusing for many of my students. So it probably be the same for you guys. So yeah, I just spent quite a bit of time on explaining this. So if you don't understand it, or if you're still confused a little bit, just tell me. Like, leave a comment, and I'll try to find another way to explain it in better way. Okay? Sweet. Number 12. 4 test tubes are set up as shown. Which test tube contains the least carbon dioxide after 1 hour? So this can actually become quite confusing. So if you look at number A, okay. So if you look at number A, you can see both animal and plants are present. Yeah, animal and plant are present. So, which means they can respire because they're living things. They both respire to produce CO2. However, plants should photosynthesize and use carbon dioxide for the photosynthesis. But in this case, it's actually covered by black polythene to keep out the light. So the plant cannot photosynthesize, so it's not going to use CO2. So overall, there's going to be a lot of CO2 in A, test tube A. For B, there's going to be slightly less CO2 than A because there's no animal, but still this plant can no longer photosynthesize, but it's just going to respire, so it's going to produce a little bit of CO2. But for C, there's a plant and there's light, so it's going to photosynthesize quite a lot, and it's going to use CO2 inside the air, inside the tube, so it's going to suck up all the CO2. So I guess the answer should be C, because the question is asking which test tube contains the least carbon dioxide gases. So the answer is C, not D, because animal will produce CO2 by respiration. So yeah, D will produce CO2 as much as B, and A will have a lot of CO2, but C will have the least amount of CO2. Oh, good. Number 13. The activity of amylase is measured in 4 parts of the elementary kernel. Which 2 parts have the most amylase activity? Okay. So, in elementary kernel, well, let's first talk about the elementary kernel. So, elementary kernel is the kernel, or let's say a very big, very long tube that starts from the mouth, and it makes up the esophagus, and then stomach, and small intestine, and then the large intestine, and the rectum and anus. Okay? So mouth, elementary kernel, not elementary kernel, my bad, esophagus, stomach, small intestine, large intestine, rectum, and anus. So all these organs are collectively called as elementary kernel. And in the elementary kernel, there are a few parts where amylases are present. So, for instance, mouth and small intestine, especially the duodenum, which is the entrance of the small intestine. And that's it. So, mouth and duodenum is the answer for this one. The colon is another term to describe large intestine. To be more precise, it's a part of the large intestine, but in IGCSE, they often use colon to refer large intestine. So keep in mind that, okay? Or keep that in mind. Not keep in mind that. Yeah, keep that in mind. My bad. So stomach does not contain amylase. It contains pepsin, which is protease. That's the enzyme that breaks down the proteins. Amylase is an enzyme that breaks down the starch into maltose, or sometimes you can just say glucose. But strictly speaking, amylase enzyme breaks down starch into maltose, and then maltase breaks down the maltose into glucoses. So that's the proper way of describing the starch digestion, but it's not actually 100% proper because starch doesn't get broken down by amylase alone. But in IGCSE, you guys don't have to know that. So, okay, so amylase is present in mouth and duodenum. In mouth and duodenum, which is the entrance of the small intestine. And that's it. Okay, so the answer is C for this one. Number 14. Biological washing powder can be used to remove stains on clothing. Which enzymes will remove stains caused by starch, fat, and protein? So, okay, this is talking about the enzyme. So, biological washing powder contains many enzymes. I mean, not that many, but there's quite a few. And it says the enzymes will remove starch, fat, and protein. So you have to find the enzymes that's going to break down starch, fat, and protein. So starch gets broken down by amylase, like I said before in question 13. And fat will be broken down by lipase. And protein will be broken down by trypsin. Yeah. Well, there are two proteases or two enzymes that can break down the protein. One is trypsin, which is present in small intestine, which is actually made by pancreas but present in small intestine. And another one is pepsin, which is present in the stomach. Okay. So trypsin is made by pancreas and then it gets stored in small intestine. And pepsin is produced in stomach and also gets stored in stomach. So anyways, the answer is B for this one. Number 15. The graph shows the rate of uptake of magnesium ions by two similar plants X and Y. The roots of each plant were placed in a range of solutions. Each solution contained a different concentration of magnesium ions. All other conditions were kept constant. The question is, what is a possible explanation for the difference in the results for the two plants? So, within the roots consist of cells called root hair cells. Okay, and on their membranes, they have many protein molecules. Some of them are called channel proteins or protein channels. The function of these proteins are to allow the movement of particles from outside to inside when there's a lot of them. So it promotes a diffusion. So diffusion is the movement of particles from high concentration to low concentration. Okay. And these proteins allow substances or particles such as magnesium ions to transport inside or to inside the cell. So plant X apparently can take quite a lot of ions, even if the concentration of the magnesium ions, like if the ion concentration gets increased. but it does reach some limitation here. But Y, plant Y, looks like it can't take that much, which means that it may lack number of these protein molecules that allows diffusion to occur. So I would say A is the good answer here. Okay. Oh, good. Number 16. What will increase the rate of transpiration in a plant? Okay, so what will increase the rate of transpiration in plant? So transpiration is, let's talk about transpiration first. Transpiration is the loss of water vapor by evaporation going out of the leaf, going through the holes called stomata. There are many pores or holes called stomata underneath the leaf, usually underneath the leaf. And there are 3 factors that can affect the rate of transpiration. First one is temperature, of course. Second one is humidity. Third one is wind speed. Okay. So as the temperature goes up, there's going to be more evaporation occurring. So there's going to be increase in transpiration. Before humidity, if there's an increase in humidity, then there are a lot of water vapors outside. So increase in humidity means increasing the number of water vapors outside. So since water vapors are gases, now relatively the water vapors inside the leaf is less than the outside. So gas does not travel through low concentration to high concentration. So there's going to be less transpiration occurring if increase in humidity occurs. If there's a decrease in humidity, of course, then there's going to be more transpiration because diffusion will occur. Okay, there's more water vapors inside the leaf, so it's going to diffuse more if there's a low humidity. So, and also wind speed. If there's an increase in wind speed, there's going to blow these water vapors outside the leaf, so there's going to be less water vapor outside the leaf, so it's going to move more out. It's going to move out more, not move more out, move out more. So there's going to be increase in the transpiration as well. However, I don't think A is the answer, or B, or C, or D, but let's have a look at the B. Let's have a look at B here. An increase in the surface area of the cell surfaces inside the leaf. That looks quite promising to me, because if there's an increase in the surface area inside the leaf, then there's going to be more water vapors or more water molecules present. So if you look at the structure of the leaf, there are palisade cells, there are spongy cells, and there's lower epidermis and stomata, created by guard cells. So these are palisade cells, these are spongy cells, lower epidermis, upper epidermis, and these are guard cells, and this hole is called stomata. So if the surface area of the cell surfaces inside a cell gets increased, which means these cell surface gets increased, then there are more water molecules present. So It's going to increase the amount of water vapor created, and it's going to diffuse out more because there are more water vapors, like a higher concentration of water vapors. So B does look quite promising to me. I think B is the right answer for this one. I said B, okay? Number 17. Which pathway is taken by blood in a fish? Okay, so it's talking about the circulatory system in fish. Fish has a single circulatory system. Okay, single circulatory system, and the pathway travels from the heart to the gills, and then it goes through the body, and then to the heart again. Okay, so the blood travels, it enters the heart, and then goes to the gill and gets oxygenated, and then travels through the body and provides all the oxygen and nutrients, and it goes back to the heart again. So heart, gills, body, and then to the heart again. So the answer is C. Okay. Number 18. The diagram shows a section through the heart and its blood vessels. Which row gives the blood vessel name and the direction of blood flow in vessel X? Okay, so X is located in upper part of the heart. So automatically, I think this is an artery. There's a type of artery. And when you look at the heart diagram, your right side is always the left one, and then your left side is always the right side of the heart. So the right side of the heart receives the blood through a vein called vena cava, and it enters the atrium and then ventricle, and it goes out of the heart by an artery called pulmonary artery, because the term pulmonary means the lungs, and artery always transports the blood from the heart. So which means the blood will now go to the lungs. Pulmonary means lungs. So artery means the blood is pumping, or the blood is transported from the heart, so from the heart to the lungs. So X is pulmonary artery, and it's moving, the blood is now moving towards the lungs. So the answer should be B. Okay. Number 19. When a pathogen enters the blood, the immune system uses different mechanisms to destroy the pathogen. The diagram shows one of these mechanisms. Which row describes the structures involved? So this is pathogen. On the surface of the pathogen, there's always an antigen. So Y is antigen, so the answer should be either C or D. And X is something that binds to the antigen, so it must be antibody. And X is made by the cells called lymphocytes. So phagocytes are the cells, the immune cells, that engulfs the pathogens, which means it ingests or it consumes the pathogens. So the word phago means to eat, and cyte means cells. So cells that eat is phagocytes, or are phagocytes. The lymphocytes produce antibodies to protect our body. So the answer should be C. Number 20. What is the approximate percentage of oxygen contained in the air breathed out of the lungs? So, this is something you guys should memorize. So in the air, there's about 20 to 21% of oxygen. And there's about 0.04% of carbon dioxide. But when you take in the air, like these stops, and when you breathe out, the concentration or the percentage changes to 16% in exhaled air, and the CO2 changes to 4% in exhaled air. So now it's asking about the oxygen in the air that breathed out of the lungs. So the answer should be C. Okay? So this is just something that you guys always should memorize or remember. Okay? Inhaled air, and this is exhaled air. All good? So make it as a table, like this. Like that. Okay. Sweet as. Oops. Oopsie. Maybe like this. Yeah, something like this. Oh my god. I'm gonna give it up. Okay. You guys know what I mean. Alright. Number 21. The diagram shows an experiment to investigate the respiration of yeast. Oil prevents oxygen entering the glucose and yeast suspension. If no oxygen is present in the glucose and yeast suspension, what will occur? So this question is asking what happens if there's no oxygen? What happens to the yeast when there's no oxygen, but only glucose? So the thin layer of oil is present to prevent oxygen from going into the yeast suspension. So it's keeping it anaerobic, and during anaerobic respiration, glucose gets converted to ethanol and carbon dioxide in yeast. So ethanol will be produced, yes. So it's either A or B. And it's also going to produce CO2, and when CO2 goes into the lime water to react with it, it will go cloudy. Okay? It will go cloudy. So this is something that you guys just should remember. There's another indicator that will show the presence of CO2. It can actually show how much CO2 is present, not precisely how much, but if there's it shows how if there's many carbon dioxide molecules or less. The indicator is called hydrogen carbonate indicator. The hydrogen carbonate indicator is initially, it's kind of, let's say, kind of reddish, like it's like pink or red color. And if there's much CO2 present, it changes into yellow color. If there's less CO2, it changes into violet or purple color. Okay, so here's another indicator or solution that can be used to predict or see or see how much CO2 is present. Okay? Anyways, the answer for this question is B. Number 22. Which row correctly completes the balanced equation for aerobic respiration? This is easy peasy. So aerobic respiration require glucose, which is C6H12O6. So, the answer is B straight away. And of course, products are CO2 and water, and you need 6 moles of water. Okay, so the answer is B. This is an easy question, very easy. Number 23. In a healthy people, which substance is completely reabsorbed into the blood from the kidney tubules? The keyword here is completely reabsorbed. So answer is A, because our body just loves glucose molecules. We don't want to waste even a little bit of it. We don't want to waste it. So we don't want to excrete it, or we don't want to get rid of it. So we completely reabsorb them all. The only times that we don't completely reabsorb them is having if there's a lot of glucose molecules inside our body due to diabetes or kidney failure, or like gallstones produced, then you can't completely reabsorb all of them, and that can actually become quite a bit of problem. But normally, we completely reabsorb all of the glucose molecules that are inside our body. Okay, so the answer is A. If we completely reabsorb all the salts, then the water potential inside the body will decrease, and that can cause a bit of problem as well. Urea is not really needed in our body. I mean, it gets reabsorbed like tiny bit, but never completely reabsorbed. And water, same thing. So if the water is completely reabsorbed, then our water potential inside our body will increase, and that can cause problems. And also, we can't excrete the urea out of our body. Urea with excess water and excess salt create urine. So putting water in urea or together with urea is the way that we get rid of the urea or excrete. And without water, that can't happen. So we can't completely reabsorb all of that water. We have to excrete some of them. Okay? So the answer is A for this one. Number 24. The diagram shows a synapse. What are the labeled parts? So, synapse. W looks like a vesicle to me. So, vesicle containing neurotransmitters. So, vesicle. So, the answer is either C or D. And X are neurotransmitters. So, the answer should be C. But let's have a look at the others. Y is called synaptic cleft, where neurotransmitters gets concentrated or diffuse into. Z is the receptor where the neurotransmitters binds to, or neurotransmitters bind to. So the answer is C. Okay. So I'll just explain a little bit more about this one. So when a neuron sends out an impulse and gets transmitted, when this impulse gets transmitted to another neuron, what happens is that the junction between the neurons is called the synapse. So as the impulse arrives at the end of the neuron, it reaches the synapse, and then this impulse will trigger vesicles to move and fuse with the membrane. And it's going to release the neurotransmitters inside, and these neurotransmitters will be concentrated, or they will get diffused into the synaptic cleft, which is a space between the synapses, or synapse, presynapse and postsynapse. So these neurotransmitters will bind to the receptors on postsynapse, and that's going to cause another impulse to be transmitted or to be made. Okay? So that's how the impulse is transmitted between the neurons. Okay? Cool. So the answer is C for this question. Number 25. Which row shows the actions needed for the eye to focus on a distant object? So, in order to see a distant object, the lens that is present inside the eye should become thinner. So, when we think of the lens is connected to the muscle called ciliary muscle. Ciliary muscle is rather circular, and the lens is connected to the ciliary muscle by ligaments called suspensory ligaments. So when the lens has become thinner, it has become stretched. So the muscle relaxes and gets expanded, and it pulls the lens by pulling the suspensory ligaments. So now suspensory ligaments is pulling the lens in many different ways. So it causes the lens to become stretched. So it becomes thinner. So it was initially like this, but if it becomes stretched, the lens becomes thinner. Right? So the ciliary muscle has to relax, and of course the lens has to become thinner, so the answer is D, because suspensory ligaments become tightened because it gets pulled. That's why the lens becomes thinner. So the answer for this question should be D. Number 26. The liver and the pancreas work together to control the concentration of glucose in the blood. Which statement is correct? So, the liver and the pancreas work together. And it says they control the concentration of glucose in the blood. How do they do so? So first of all, the liver converts glucoses into glycogen. Of course, it happens in the presence of a hormone called insulin. And also the other way around happens as well. Glycogen, which is a polysaccharide that's formed by many glucose molecules. And glucose molecules are like this. So glycogen, big chunk of glycogen gets converted back to glucose molecules if a hormone called glucagon is present, or when glucagon is released. So for this one, I would say the answer is D, because the pancreas responds to fall in blood glucose by increasing the release of the hormone glucagon. So when there's a fall in blood glucose, so when there's less glucose, then the pancreas releases a hormone called glucagon, and then the glycogen gets converted back to the glucose molecules in the liver. Okay? So the pancreas is the organ that produces these two hormones, glucagon and insulin. And insulin is produced when there is an increase in glucose concentration in the blood, and the glucogen gets released when there is a less, or there's a decrease in glucose molecules in the blood. So insulin is released in order to change glucose molecules to glycogen, thereby decreasing the blood glucose level. And glucagon is produced or released if there's less glucose molecules in the blood. So what glucagon does is that it converts glycogens back to the glucose molecules, thereby increasing the blood glucose level in the blood. Okay? So therefore, the answer is D. Number 27. The statements are about the use of antibiotics. Which practices will increase the chance of the development of antibiotic resistance in bacteria? So let's talk about antibiotic resistance in bacteria. So some of the bacteria can develop antibiotic resistance, which means it doesn't get killed by antibiotics. So they're, I suppose we could say they're immune to it, or better say they're just they have resistance. So they're okay with the antibiotics that usually kill them. How do they get formed? Usually by mutation. But how does this happen? Of course, mutation can happen by all sorts of things, like usually by UV lights, like any sort of irradiations, and mutagenic substances. So mutagenic substances means the substances that can cause mutations, like chemical substances. Some of the chemical substances can actually give tremendous amount of stress to the cell or organism, and it can alter the gene sequence, thereby mutating the gene sequence of the organism. So how does this happen? So let's have a look at number 1. It says using an antibiotic to prevent infection. Well, it's quite vague. It says to prevent infection. There are all sorts of infections. So infections can occur by bacteria. It can also happen by virus or viruses as well. So it's, in a way, it's quite vague. So if you just use antibiotics to prevent infection, regardless of the cause of the infection, then it can actually develop the antibiotic resistance in bacteria, because if the infection happened not by bacteria but virus, then the bacteria inside the body will be exposed to antibiotics that can be effective to them, but not enough. So some of the bacteria can survive, and they can become antibiotic resistant bacteria as well. But not only it doesn't actually happen just by the exposure to antibiotics, but some of them may live, and then they can divide themselves by binary fission. And then during the fission, some of them can actually become mutated. Maybe one of them can become mutated. after the exposure to antibiotics. So this becomes antibiotic resistant, and this one, although this one dies, this one will live, and then they will just increase their numbers. So anyways, back to this question again. So preventing infection is quite vague, so I would say this cannot be the answer. But because if you look at number 2, it says using an antibiotic known to kill the bacteria causing an infection. Yes, antibiotics can kill bacteria. Okay. So we should only use antibiotics when the cause of the disease is identified. And if it's caused by, or if the disease is caused by the bacteria, yes, then you should use antibiotics. So, okay, this procedure of using antibiotics to kill bacteria that causes an infection is, of course, right way to go. So, I'll say this will not cause the antibiotic resistance. So number 3, using an antibiotic only when essential. Yes, this is absolutely crucial. It's absolutely right way to go. Yes, you should, or we should always use antibiotics when it's totally essential. Okay. Number 4, using an antibiotic to kill a viral infection. No, antibiotics only work on bacteria. Okay, antibiotics only work on bacteria, not on virus, never. So 4 and 1 are like big no-nos because they will increase the chance of development of antibiotic resistance in bacteria. Hence, the answer should be B, 1 and 4 only. Okay? Number 28. Which description of cross-pollination is correct? So, cross-pollination happens when the pollen grain from one flower moves to the stigma of another flower. So pollen grains are usually present in a structure called anther, and it arrives in the part called stigma. So anther is the male part of the flower, and stigma is a part of the female part in the flower. So this part is called style, and this is called ovary, and inside ovary there's an ovule and egg and stuff like that. So the anther produces pollen grains, and they usually transport by wind, insects, or any other animals. Okay, so if this happens, that's called cross-pollination. If the pollen grains from the anther just travels to the stigma in the same plant or in the same flower, that's called self-pollination. This is self, and this is a cross-pollination. So hence the answer is A. Okay. Number 29. The diagram shows a sperm cell. Which part contains enzymes that digest the jelly coat of an egg cell? So you're looking at sperm cell. The sperm cell consists of 3 parts: head, midpiece, and the tail. Okay, so within the head there are 2 main parts called acrosome and the nucleus. It's this acrosome that contains many hydrolytic enzymes, or you could just call it digestive enzyme, that dissolves the jelly coat of an egg when it makes a contact. With the egg. So the enzymes inside the acrosome, they dissolve this the jelly coat, and then they insert the nucleus into the egg, and that process is called fertilization. So for this question, the answer should be D. Number 30. Which cell contains a haploid nucleus? So haploid nucleus is present in specific cells, and they are collectively called gametes, or in other words, they are sex cells. Okay. So amongst these four options, there's only one sex cell, which is of course the sperm cell. Neuron is a body cell, so it should be diploid. Skin cell is also a body cell, so it should be diploid. And red blood cell does not have nucleus. So only gamete in here is the sperm cell, so the answer should be B. Haploid means it has a singlets of chromosomes, not in pairs. So chromosomes are not present in pairs, but singlets. But diploids are pairs. So for instance, the humans, we have 46 chromosomes in each cell, in each body cells. There are 46 chromosomes because the chromosomes are in pairs. So we've got 23 pairs, thereby 46 chromosomes. But for gametes, like sperm cells and egg cells, they have only 23 chromosomes, not in pairs, but 23 chromosomes. And they are called haploid. Okay? Number 31. The diagram shows the chromosomes of 4 daughter cells produced by meiosis. Which parent cell produced these cells? So, meiosis, huh? Meiosis is a cell division for sex cell production. So when we make sperms, when humans or animals make sperms or eggs, the cell divides twice to make 4 daughter cells, and they are genetically non-identical, so they are genetically all different. They can be either sperms or they can be eggs, but to be more strict, meiosis only produce 1 egg cell as a product. So which parent cell produced these cells? These cells, the products of meiosis, are haploids, which means they're not in pairs, they're in singlets. So the parent cell was initially diploid, and then it becomes 4 daughter cells with haploid nuclei. So you just have to find a cell that contains twice as much as chromosomes inside one of those 4 daughter cells. So hence the answer should be B, because there are 4 chromosomes. they have 2 chromosomes each, and the parent cell must be diploid, so it should have 4 chromosomes. Hence, that the answer is B. Number 32. When damaged tissues are repaired, cells undergo division by... So it's asking about the type of the cell division that happens when damaged tissues are to be repaired. So it's typical, the typical cell division that does the repairing is, of course, mitosis. This is because mitosis produces 2 identical daughter cells. So it forms clones, thereby replacing or repairing the cells. So answer should be either C or D. And just like I said before, the mitosis produces genetically identical cells, so the answer should be C. The genetically different cells are formed by meiosis, not mitosis, okay? Number 33. The graph shows the masses of 2 different types of tomato. What can be concluded from the graph? So, looking at the graph, you can see this type of graph is called histogram. It looks like bar graph, but it's actually histogram because the x-axis is showing the bar, and it's actually in a range between 10 to 20, and the next bar lies between 20 and 30, and so on. So it's not typical bar graph, but it's histogram. So what can we conclude from the graph? So it's showing the type 1 tomato is showing continuous variation. Is correct. So the continuous variation is the variation that's that can be let's say expressed in using numbers. So for continuous variation, there are many intermediate values. So for instance, like mass, there's not only just 20 kilograms or 20 grams or 40 grams or 60 grams, but also there are 21 grams, 22 grams, and there are even like 21.12 grams, 21.213 grams, and so on. So there are many intermediate values in between the intervals, and these all different, let's say, characteristics, they can be considered as characteristics or features. They are called continuous variation. So there are tomatoes that weigh like 21.23 grams, and there are also tomatoes that weigh 21.5 grams. So that's there's a huge difference between the continuous variation and the discontinuous variation, because discontinuous variation is very, very discrete. So for instance, blood type or blood types, there are like A, B, and O types, right? For the blood types, there's no such thing as A, B, O type. There's only discrete blood types for the blood types, and they are called discontinuous variation, like tongue rolling as well. For tongue rolling, you either can roll your tongue, or you cannot. There's no such thing as intermediate between yes and no in terms of tongue rolling. So this is showing a continuous variation because these number of tomatoes lie between 10 to 20 grams of weight. This one belongs to, or it lies in between 30 to 40 grams. So type 1, actually type 2 as well, they both show continuous variation. So B is right because type 1 indeed showed continuous variation. Okay? Number 34. Which adaptation may be present in a xerophyte? Xerophyte, the word phyte means plants or plant, and the word xero means dry. So xerophytes are the plants that live in very dry area. And in this case, we're gonna talk about the marram grass. Okay, marram grasses. They are plants that have rolled leaves. So the rolled leaves have hairs inside, and these hairs prevent the water vapor from going out of the leaf, so they stay inside. They stay inside the leaves. So this is how they conserve the water. So yep, the rolled leaves is one of the adaptation as well, and they have hairs. So C is the right answer for this one. Let's have a look at A, leaves with small surface area. Okay, yeah, that can be a adaptation as well, but large numbers of stomata, no. If there are a lot of stomata, then it means there are more pores that the water vapors can escape. So if there are many stomata present in the leaves, then more water molecules will go out and they'll lose more water. The plants will lose more water. So A is not the answer. B, little or no xylem tissue and leaves with large surface area. If the leaves have large surface area, then there are more spaces or more surface for the water molecules to go out, like escape from the leaves. So no. Number D, thin or no cuticle and deep roots. Deep roots is, it's absolutely right. It's a very good adaptation that the xerophytes can have. However, thin or no cuticle, no. Cuticles are like waterproof materials. So if the leaf has no cuticle, the water molecules inside the leaf will just evaporate and they'll just get out of the leaf. So no way. So the answer is C for this one. Number 35. The diagram shows a food chain in a rock pool. What will happen if the number of secondary consumers increases? So the food chain starts with seafood here, and it gets eaten by whelks, and gets eaten by crabs, and gets eaten by seagulls. So it's a food chain that shows a producer, primary consumer, secondary consumer, and then tertiary consumer. So if the secondary consumer, which in this case is crabs, they increase, then there are more crabs that eat whelks, and if they eat more whelks, the number of whelks will decrease, and then the seaweeds will increase, and the seagulls will increase because seagulls will eat crabs, and they're more food now because the secondary consumers increase. So the answer should be C, fewer whelks. Okay, cool. Number 36. What is defined as all of the populations of different species in an ecosystem? So in order to understand the meaning of this, it is important to say or talk about species, the term species. So species is an organism, or let's say it's a group of organisms that have similar characteristics, and they can give to a birth to fertile offspring. So they can interbreed and give birth to a fertile offspring. So if same types of species gather around, then they can form a population. So population is a group of same species in a same area, in a same given time. So if different populations come together and gather around, it forms community. Or they form community. Okay. So number 36. All of the populations of different species in an ecosystem must be community. It says all of the populations is plural of different species. Okay. So let's say A. So there's a species called A, and if many A's come together, they form a population. And if many populations come and gather around together like this, then they form community. So this is a community. Okay. Number 36. What is the role of anaerobic respiration in bread making? So the anaerobic respiration that you learn in IGCSE biology happens in animal cells or animals, and in yeast as well. So when it talks about the bread making, it must be happening within the yeast. Okay. So the anaerobic respiration that happens in yeast involves glucose molecule turning into ethanol and carbon dioxide. So the carbon dioxide that is formed, it inflates the bread dough. So let's say this is a dough, and if there are like yeast in there, without the oxygen, they're gonna do anaerobic respiration, and they will form CO2. So CO2 will be given out, and the whole thing will expand. So yeast is used to make bread because they form CO2, the gas, to make the bread rise or expand. Okay, that's why we see like all sorts of like many holes within bread. Okay, so 37 is B. All good. Number 38. A crop plant has been genetically modified to make it resistant to herbicides, which is a possible disadvantage of introducing this new crop plant. So this question is talking about genetic modification, the downside of the genetic modification. And of course, the crop plants can become resistant to herbicides, so we can just freely use herbicides to kill weeds However, there are actually some downsides of this technique or technology called genetic modification. A is not yet right answer because loss of weeds, reducing competition, is not a downside. It's not a disadvantage. So if there's less competition, then the crop plants can receive or they can absorb more nutrients. So it's a good thing, right? So A is not the right answer, but B, some weeds might become resistant to the herbicides. Yes, this is the downside of it. So using too much herbicides can actually promote some resistance in weeds as well. So if the weeds become resistant to herbicides, then it's going to be harder to kill them. So A is the right answer, but let's just go on. Let's have a look at C. The crop plant is unharmed and produces a higher yield. This is a good thing, right? So it's not a disadvantage. So it's a good thing. So it can be an answer. D. The new gene will appear in the new generations of the crop. So this is very vague. We're not sure whether this new gene can actually become a good thing or not. So this is very vague. So we can't say that's the answer. Hence the right answer for this one is B. Number 29. Which process in the nitrogen cycle is involved in the breakdown of amino acids in living organisms? So, let's talk about nitrogen cycle. When the living organisms die, then their proteins become broken down into amino acids. And these amino acids, they get converted to ammonia or urea by the process called deamination. Okay? So converting amino acids to ammonia or urea is called deamination. Hence the answer is A. B is not the right answer because decomposition is just basically means decomposing the dead organism. So when the dead organisms become decomposed, it's going to be broken down into protein or amino acids, and that's it. But this question is asking about the breakdown of amino acids, so can be the answer. It cannot be the answer, and C is not the right answer because denitrification is converting the nitrates into the nitrogen gas. That's called denitrification. And digestion is not involved in the nitrogen cycle, so obviously it's not the answer. Okay, and this question's answer is therefore A. Question 40. When a river is polluted by fertilizer, the following processes may occur. What is the correct sequence for these processes? So, the river pollution, the water pollution that's happening in the river by fertilizer. So, if the river is polluted by fertilizer, eutrophication will occur. Okay? Eutrophication is a process that starts with fertilizers entering the water source. And if the fertilizer gets dissolved in the water, then the plants, aquatic plants, will just love it. And they'll grow even more. However, aquatic plants underneath the water are not the only ones that will be happy about this. There are other plants like algae that will obtain or consume these fertilizers and grow more. And it's going to eventually block the sunlight that's entering the water. Since these plants cannot absorb light energy, they can't photosynthesize, and they'll die. If plants die, then the decomposers will decompose these aquatic plants. So, since decomposers are also organisms that will use up the oxygen within the water, usually aquatic plants will supply the oxygen into the water, but since they're dead, there's nothing that can provide oxygen to the water. So there's a decrease in oxygen level, and the other fish and organisms will die because simply there's no oxygen. And more decomposers will decompose these dead organisms, and they'll use even more oxygen, and later there's going to be no oxygen in the river or lakes. So if we identify the order of this process, then the increased growth of producers is correct. It's the first one. And then the increased aerobic respiration of decomposers is the next one. And then decreased oxygen concentration in the water is the final one. It's the last one. So C is the answer for number 40. Okay? Hope you guys had fun with these questions. And if you have any questions, just leave the comment and below. And if you guys can subscribe to my channel, that'd be wonderful. It will help me a lot, because it does actually take quite a long time to make these videos. But with your help, I'll be more than happy to make more of it. Okay, so all the best, and just leave me any comments that you guys want to ask, and I'll see you guys next time. All right, see ya.